This is the Anthrobiology Podcast, hosted by me, Gabby LaPera. Every 2 weeks, I interview a biological anthropologist about what makes us, us. Hello, everyone. Welcome to another episode of the Anthrobiology Podcast. Joining me today, I have Dr. Erin Blankenship-Sefczek of Creighton University. Hello, how are you?
Good. How are you?
I'm really good, thank you. I'm really excited to have you on because I actually met you in person at AABA, and that was my first time at the conference, and you were so nice, and you're so smart. I'm very happy to have you here.
Oh, thank you. I'm super excited to be on. This is my first ever podcast, so I think it's a good introduction. I'm excited to talk more. And I very much enjoyed it's such a small world in anthropology, so it was fun that we made connections with other people, and then you knew people that I was staying with. So, it's just a fun community.
Yeah. Because Dr. Jesse Goliath, who was my first episode this year, did you guys go to grad school together? Is that right?
We did. Yes.
Yeah.
We went to where he was a couple cohorts ahead of me at Ohio State. And we actually met at an AABA conference in Portland before we were going to be my husband and I were going to go to Ohio State. We knew we were going, but we hadn't been there yet. And so we met him to talk about some stuff and, the program. And we've been best friends ever since.
It's just so nice. Sometimes people write to me and they're like, "Are your guests as nice as they seem?" And I'm like, they really are. I met them all in person and they were like, I was just some rando podcaster and everyone was so nice to me.
It's a good community. It really is. Yeah.
Yeah, totally. Well, I'm really excited to have you on. I believe that you are the first tooth person this year.
Yay!
Last year was the year of teeth, so
I'll just continue it. You can sprinkle it in.
I think I had like 4 or 5 tooth episodes last year. Like it just like the hits just kept on coming. So for everyone who's very excited about mouths, another episode for you. So can you tell me a little bit first about what your like major area of research within teeth is? Like what do you focus on?
Sure. So right now, the trajectory of my research is looking at the size and shape of teeth in the presence of developmental disruptions. So I'm really interested in looking at, enamel defects, and those are not, the etiology is non-specific. So could be malnutrition or disease. So when those are present, what is that doing to the adult tooth? Really, it's an approximation of during development. So what's happening with the cell signals and the tissue interactions, but what's the then phenotype that we're seeing? And specifically looking at spaces between cusps and then any extra or accessory cusps. Right now, I'm looking at the upper teeth more than the lower teeth and looking at like the cusp 5 and the Carabelli's cusp. So I want to know if in the presence of an enamel defect, does cusp spacing change? And then does the presence of an accessory cusp also change?
Okay, we're going to back up a little bit just because it's a lot of technical words. First, just for listeners who are new to the show, what does phenotype mean?
Oh, the phenotype is what it looks like. Yeah. The outward expression.
Yeah, perfect. So like you're looking at like, because I think that what a lot of people don't think about when they think about genetics is like they think that you just have your genes and they're expressed and like it's like a 1-to-1 ratio, but like you can have genes that don't get expressed, and so the visual component looks different. So for example, what are the ones that they always teach in like intro to bioanth classes where like you have like the super simple ones where like if you're homozygous you're one way, if you're heterozygous. Do you know what I'm talking about?
Yep. It's like if you put your hands together and one thumb over the other one is one of them. Dimples are one, attached earlobes, all of those. Yeah.
Yes. The hand one that's always crazy to me. So like listeners, if you take your hands and just like interlace them, look at the thumb that's on top. Mine is left.
Mine is right.
Yeah. It's super, but it's one of those things that you wouldn't expect it to be genetically controlled, but it is. Or like your earlobes, the attachment, what you're looking for is like some people have like distinct lobes and then other people have it. It just kind of like melts into the side of their head. Not that makes it sound unattractive. It's not. It's just a different. But that's the phenotype. Like, is it touching or is it like a little round lobe next to it?
Yeah.
And those have like kind of like more simple gene controls. So like if you have the dominant trait, even if you have your genes always have two, right? Because you have one from mom and one from dad. So depending on whether the gene is dominant or negative, recessive, not negative. Jesus Christ. Dominant or recessive. If you have a dominant gene, that's always going to express. If you have a recessive gene, you need to have two of the recessive genes for it to be expressed. I don't know if that was a great explanation of genetics, but if it wasn't, feel free to write to me and tell me, and I will very happily explain it to you in a better, more concise way. But yes, so you're looking at like the actual expression of the genes, the phenotype, like what things actually look like. Can you explain about cusps? Because I don't know that a lot of people have like looked at molars.
Sure. I know it's hard. Sometimes I'm in, I do this, so I'm in my own head and everybody knows what's going on, right? So on your tooth surface, and this is true of all the teeth except for your the anteriors, which are your incisors and canines. So when you smile, those are the ones you see, is the big incisors in the front and on the top, and the slightly smaller ones in the front on the bottom. And then your canines are the really pointed ones right next to those. You have 4 incisors and then a canine on either side. And so your premolars and molars, which are the ones in the back, if you run your tongue over them, you can feel little peaks on the surface. Those are all your cusps. And your premolars usually have 2, and your molars, the upper molars, usually have 4. We call those the principal cusps. So those are the ones that almost always form. I say almost always because there are exceptions. You can have molars with 3, upper molars with 3 cusps. But those are the ones that are pretty standard and develop in most people. And then in the bottom, you usually have 5. And then any extra or accessory cusps is what we would then consider ones that develop after or are dictated by the development of those principal cusps. And so you can have them on the surface between those cusps that form first, so those principal cusps, or around the outside of the tooth. The ones on the surface we call occlusal accessory cusps, and the ones around the outside would be peripheral accessory cusps. And so the carabelli is a really common one in upper molars that forms on the tongue side, so on the inside of your mouth. And then the cusp 5 would be between 2 of those principal cusps towards the back of the tooth. And so those are basically what I'm looking at, is the spacing of those cusps that form first, how they influence the formation of any extra cusps that are there. And that's where there's a lot of, there's a movement, there's more evidence showing that the presence of stress markers or physiological stress during development, like malnutrition and disease, change the space of those principal cusps, or the ones that normally form or usually form. And then the spacing of those dictates whether any extra cusps can form. And the reason that's really important to understand is because those cusps, the size and whether or not they form, is how we look at biological affinity or relationships between populations.
Yeah. I mean, when I was coming up, people would like take it as like an indication of like potential ancestry. But if what you're saying is like, if like it can happen because of developmental stress, then like maybe this isn't like a great trait to use to make those kinds of determinations.
It's more complicated than just a trait presence equals that ancestry. That's my biggest research question, really, is what's influencing the presence of these accessory cusps. And because we use, and yes, there is definitely a genetic component to it, it's also important to recognize that it's a really complicated structure. The tooth is you have, during development, you have tissues that interact with each other. They overlay, and there's signals physiologically. There's signals that go back and forth that tell one tissue to do something where like cells differentiate, and then you have one portion of the tooth, and then you have cells signaling that tells these other cells to do these other things. And so it's a really complicated interaction. So strict genetic control is not really the real interaction that's happening, the only one. If the individual has a disease or malnutrition is involved, then those, just like other structures or other systems in the body, they're going to get disrupted. And we see that. I mean, that's why we see hypoplastic defects. Hypoplasia refers to less matrix or less of that tissue. And then, so in the teeth, that hypoplastic defect a lot of times is seen as a line. So we call that linear enamel hypoplasia. It could also be pits or discoloration, where there's a change in the amount of that tissue there. So we know it happens in teeth. Why wouldn't it also then happen for these cusps that form?
Right. Yeah. So it's not just affecting the enamel. It's also like, especially if it happens at a crucial developmental point, it can affect the actual, maybe structure is the wrong word, but the actual morphology of the tooth.
Exactly. Yep, exactly. And that's what my research really centers around is, if there is the presence of stress, what is that then going to do for the tooth? And there's this idea, because modern humans have simplified less ridges and extra cusps, and the numbers and the size of teeth, it's all smaller. And so there's kind of this idea that with a smaller tooth, there's less space on that surface for anything else to form or for extra cusps to form. And so this is where the cusp spacing comes into play, because it's not just about the size of the tooth. It's also on molars, it's also about how those cusps are spaced on that surface and whether or not an extra cusp has the potential to form.
When you say modern humans, you mean like compared to humans, like anatomically modern humans that are like 10,000 years ago, or do you mean
both? So anatomically modern humans, even, early on in anatomically modern humans, we see a reduction in tooth size. And then, yes, definitely a reduction in tooth size post-agriculture, right around the transition and post-agriculture. So we see this real continual decrease in tooth size and a real increase in the rate of smaller teeth with agricultural populations. And then even earlier than like the beginning of anatomically modern humans, we have different morphology to some extent than even Neanderthals or earlier hominins. So we're seeing a change in the tooth structure already, and with less cusps and less ridges and those kinds of things. And so there's this idea, again, that smaller teeth mean simplified trait expression, which to some extent is true. But that also, it has to do with the size of the tooth and the spacing of those cusps on the surface of it.
Because they all have to interact with each other. Like the area is so small, they all have to interact with each other.
Right.
This makes a lot of sense. It makes a lot of sense to me. It makes sense to me that developmentally, like that's, if you experience developmental stress, it's going to affect outcomes for your teeth. I think we should probably mention when are teeth like developing? Like when would the stress be happening that would be affecting the morphology?
Sure. So a lot of your deciduous, or all of your deciduous teeth, are developing during the gestation period. So during pregnancy, that's when those teeth are starting to form. And then you have a really early development of your first molars. So the initiation is going to start pretty early on. And then the canines are also really early. So in my work, I look at incisors too, but also canines. And on the tooth surface, from the tip of the cusp to where the enamel ends and the root starts, you can kind of estimate based on where you are on the crown when it developed. And so you can match where that portion is on the crown to the development of molars when those molars initiate in utero. And you can start to talk about the relationship between this early stress and when actually those signals were being sent to the molar.
That's really cool. It's just really neat.
And the reason why that's important is because, as with a lot of biological anthropologists doing bioarchaeology or dental anthropology, I work with past populations. and not living populations. And so I don't have an illness history for these individuals. So what I'm trying to do is a proxy for like cusp spacing is an approximation of all the cell interactions that are happening. And I can describe that more if you really want me to. But there's, it's a sort of an approximation of what's happening in that molar. And then the anterior teeth, those incisors and canines, are where along that surface, if you have a defect, where that defect falls, that's going to help tell you the age. So then you can match that up. And that's our best approximation or best guess of when those two activities were happening.
I mean, I think that's amazing. That's so brilliant that you can use the teeth to like date the stress and then look at how the stress would affect, because the developmental timeline. It's so cool.
I think it's pretty cool too.
So I wanted to ask you, I was reading your article, and one of like the main ideas in your article before we like really get into it is this idea of the patterning cascade model.
Mm-hmm.
And I was wondering if you could explain what that is.
Sure. Yeah. So this goes back to tooth development, and I talked about those principal cusps, or those cusps that form on the tooth surface. And so the idea is that not all of those cusps form all at the same time. You have your cusps that form first, and then your second and third and fourth. I'm going to talk about upper molars because that's what the paper is about. And that's what I spend most of my time, most of my research is on maxillary molars. So I'm going to talk about it in terms of 4 principal cusps. So you have, during development, tooth development, at the very start, before you have a tooth structure that looks like a tooth or even sort of looks like a tooth, you have signals in these 2 tissues that says, hey, a tooth's going to be here. And then you start to see some cell changes. And then you start to see a little bit more of those tissue changes. And then you have the formation of a little transient, so a structure that is there just for a little bit of time and then goes away, called the enamel knot. That enamel knot is a cluster, so a group of cells that are going to send signals to all the rest of those tissues to tell it what to do and where to do it. And that first cluster of cells, that first enamel knot, is will eventually be the tip of your first cusp in a molar.
Okay.
Around that cusp, around that signal, there's an area called the inhibitory zone. And that's a space that's basically saying, any other cusp don't form here. You can't be here. So the size of that inhibitory zone from that very first cusp is going to influence or determine where that next cusp can form. And then that one also has an enamel knot. We call that one a secondary enamel knot with an inhibitory zone around it. And then the third one and the fourth one, all of those have that. So the patterning is what the tooth is actually going to look like. That's your sort of genetic control. Are we forming a molar, an upper molar tooth or a lower molar tooth? Is it a human? Is it a rat? Is it a mole? Whatever. That's your pattern. So what that tooth is going to more or less look like. And then the cascade is the earliest formed cusp is influencing the next one, the next cusp, and then the one after that, and then the one after that. So it's a developmental cascade. And the size, this is where the cusp spacing comes into play, because those little inhibitory zones are telling the tooth you can't have another cusp form too closely. So if you have really big inhibitory zones, you're going to have widely spaced cusps. If you have smaller inhibitory zones, you're going to have more closely spaced cusps.
That makes sense to me. This also feels like a 10th grade math problem where they're like, if the size of this circle, like how close, it's like, it's actually like COVID. It's like, if you have 6 feet of space around you, how many people can you pack into this area?
Exactly. My husband, he's also an anthropologist. He does primate behavioral ecology. We both read each other's work. And when I get to this side of things and I start talking about the specific cell signals in the paper, he's like, you lost me, blah, blah, blah. Okay, I'm back when it's done.
That's really funny. I will say I did read your paper and I thought it was like very clearly explained.
Oh, good.
We keep talking about your paper, but we haven't said what it's called, I believe. So it is the nutritional supplementation, tooth crown size, and trait expression in individuals from Tenochtitlan, Mexico. You couldn't see Dr. Blankenship-Sefczek give me a thumbs up when I said that, but she did. For the town names, I'm pretty proud of myself. So like basically, is the PCM, the patterning cascade model, is this something that's like widely accepted? Is it something that like is controversial?
It's pretty widely accepted. So it's all based on embryonic experimental data studies that were done, in a lot of it by Yuka Yernval in the early 2000s, discussing it. And it started with mice and voles. So they're mammals, and all mammal teeth develop very, very similarly. And, we can't ethically, morally do experiments on humans. So we're sort of extrapolating that information from these other mammals to humans. But yes, it's widely accepted. And there have been a number of papers discussing this idea of when the spacing of those principal cusps, so those cusps that form early, are going to influence whether or not you have a cusp on the outside, one of those peripheral cusps like the carabelli, or if you have an occlusal cusp, an accessory cusp like your cusp 5. And so that particular paper you're talking about of mine builds off of a paper by Ortiz and colleagues that was published saying that there's different conditions that peripheral and central cusps can form on. And we absolutely agree with that. And we've added some saying that, it might be, so the idea is that peripheral cusps will form if you have more closely spaced principal cusps on that surface. And that for central cusps, they're more widely spaced because you need space between those principal cusps for them to form. And that's also two of my co-authors, Debbie Guatelli-Steinberg and John Hunter, also published a paper specifically about the carabelli cusp, talking about this. So there's lots of papers that are showing, yes, this is an accepted idea here. And so we're introducing this idea that there's variation. You might have, it's not all the cusps are close or all of them are spaced out. You might have some that are close and some that are far. And so you might have the potential for both an accessory peripheral and an accessory occlusal cusp to form on the same tooth.
Generally speaking, what would like dictate the cusps being closer together generally?
Mm-hmm. Sure. So that's going to be the size of that inhibitory zone between each one. So that enamel knot's going to say,
I meant like developmentally, what would influence the inhibitory zone to like be smaller for that enamel knot to, do you see what I'm saying?
Yeah. That's what we're not entirely sure. And that's really where I think this idea of stress is important to explore more, because we're seeing that cusp spacing is more variable. That's my dissertation is also, and that paper is part of my dissertation. And then there's a bigger part that's on an archaeological context population or set of individuals. But I found in there that the cusp spacing is more variable when stress is present. So when those markers are present, or in the particular paper that we're talking about today, it's a sample with known nutritional status. So the individuals who were less nourished, who expressed pretty severe malnutrition, were showing more variable cusp spacing compared to the individuals that weren't. So there's something about that disruption in the system that is influencing the size of that inhibitory zone. What exactly? I'm not sure.
I think it's worth also taking a second to talk about who were the people that you were studying? Because I think that it kind of sounds like you're studying live people just by the way that we're talking, and you're not.
No. No. So this particular group of individuals, it was a longitudinal study that was done in the 1960s, early 1960s through the late 80s, early 1990s, which I was not born for part of that. So no, I was not part of this longitudinal study, but a doctor and a nutritionalist wanted to know what effects malnutrition had on things like body mass index, head circumference, height, weight, that kind of stuff. And so both of them were part of the Mexican Institute of Health. And so they went to this rural Mexican village and asked if they got permission. And some of the individuals in the community were qualified for a government supplementation program where when mothers were first showing signs of pregnancy, they were given nutritional supplementation. And then once the children were born, they also received nutritional supplementation. And so what Dr. Chavez and Miss Martinez did was went into this community and documented all those changes. They also looked at the health of the mothers during pregnancy and then the children longitudinally until they were adolescents and early adults. And so that was the study that this came from. And then in the early 90s, Alan Goodman was brought onto this project because he was interested in the frequency and the differences in enamel hypoplasias between the supplemented and the non-supplemented children. And so he went in and recorded all the hypoplastic defect data and looked at illness history and did that comparison. And he actually has a paper that was published in 1991 on that. And as part of that, he took casts of the individual's teeth. And so that's what I used was the casts. I got in contact with Alan earlier in my career and in my doctoral work and just asked, this is what I'm interested in. Do you have ideas for, since you do data on hypoplastic defects, would you, is there something that you might suggest I do? And he said, actually, I have this collection that would work for this. And there is this documentation of nutritional status. And so I collected all my data on the dental casts. And I, of course, put in for an IRB just to make sure I was following protocol. And they said I didn't need one because it was cast material and it had happened long enough ago that it didn't really qualify for an IRB.
Yeah. If you had been like part of the original study and you're like, I'm going to supplement some kids, but not other kids, they probably would have been like, no way.
Exactly. And that's where this collection is really unique in that because we have all of this information on the individuals and we know to some extent what their nutritional status was. There's lots of documentation.
For listeners who might be new to the show, we keep saying IRB, and that's Institutional Review Board. And those are organizations that are responsible for making sure that humans, and there's also like a version for animals, don't get hurt when people are doing research. And this is because of a history of scientists hurting people while doing research. I shouldn't laugh, but it's like, I'm not laughing. It's like one of those like panic reactions where you're like, this is so horrible. And every time I think about it, I just like chuckle nervously. But if you want to know more about why IRBs are a thing, if you look up Tuskegee syphilis study, you will have the full horrible story to give you nightmares at night. And really appreciate the work that IRBs do, even though sometimes they ask questions that you're like, I can't tell if you read anything that I wrote.
It's here to protect the people participating in research, which is good.
Yes. It's really good. It's just sometimes you're like, I don't know what's going on here. But they do take their responsibilities really seriously, which is the most important part.
Yeah.
So you had these casts. I was wondering, like, what were some of your findings in terms of what effect did you see in people who were nutritionally supplemented and people who were not nutritionally supplemented?
Yeah, for sure. So individuals who were not nutritionally supplemented, so these would be the individuals who showed a higher degree of malnutrition compared to the other group. Those individuals had more changes to their cusp spacing and also an increase in not only the frequency to some extent, but also the size of the accessory cusps that were being expressed. And so that's where this relationship between, very interestingly, there was no difference in crown size. I looked at first molars and second molars because those were the two types. There weren't as many second molars as there were first. So most of the analysis in that paper is focused on those first molars. And so what we found was that the more malnourished individuals had, even though they had the same crown size, they had a higher frequency of those accessory cusps, both the peripheral, so the cerebelly, and the occlusal or central, that cusp 5. And the reason for that, we argue in that paper, is that there's a change in cusp distance.
So that means that there was greater cusp distance to allow for additional cusps.
Well, so we found both. That's why we this new idea because or this new potential configuration because we saw more closely spaced cusps. So you have on your tooth, we talked about those 4 principal cusps, the directions, just like you can talk about anterior being in front and posterior being behind, medial towards the midline and lateral away from it. Teeth, of course, have their own set of anatomical directional terminology. So the cusps, the mesial cusp closer to the front of the mouth, that's more where the cerebelly cusp is located because it's on the tongue side or on the inside of the mouth side of that first cusp. And so those 2 cusps on the distance closer to the front were more closely spaced. And then the distal cusps, are the ones on the back end of the tooth closer to the back side, were more widely spaced.
Yes, that totally makes sense because if you have like more closely spaced cusps in the front, the only place for an accessory cusp would be on the side.
Right.
I understand now.
And on the back end, because that cusp 5 is situated between those 2 principal cusps, so those 2 cusps that normally form, they need to be more spaced out in order to make room because that accessory cusp theoretically also has an inhibitory zone around it. And so the 2 principal cusps have their inhibitory zones, and then that cusp between them would also then have to form in an area where it's allowed to. So those 2 are more farther apart. They're more widely spaced than the ones at the mesial side or towards the front of the tooth.
So like if you imagine the 4 principal cusps, you would see kind of like a trapezoid where the 2 front cusps are closer together and the 2 back cusps are farther apart.
Yep, exactly.
Which you actually documented beautifully in images in your article. If anyone is curious and wants to go see it, it is accessible and I will put it in the show notes. This is really cool. It's just really interesting. What are you working on next? Like, what is the next step in this study?
Yeah. So that particular paper that we've been talking about focused very much on the nutritionally supplemented and non-supplemented groups because it was documented that we have something of an illness history and known nutritional status. And so the first step was to show, hey, in this known group, here's what we're seeing. Now I am using Alan Goodman's LEH data or enamel defect data in general, because LEH or linear enamel hypoplasia is just one type of hypoplastic defect. I am focusing on that particular type in this next paper because that's the most commonly used in anthropology. But now the next step is to look at, independent of group membership, supplemented or non-supplemented, I'm now looking at if there is an enamel defect present, how is that relating to cusp distance, and how is that relating to trait expression, or whether or not there's an accessory cusp that's there, because that's the application for anthropology in populations, ancient populations where there is no illness history. All we really have to look at are those hypoplastic defects. And so if we can start making a relationship between the presence of those defects in a group where we have that information, then we can extrapolate that to group populations where all we have is the hypoplastic defect.
Yeah. It's also this idea of it's like epigenetic control, like what was going on with mom then also affects the kid's teeth.
Mm-hmm.
That's also super interesting to me. I just really like epigenetics. It's just really what that whole sentence boils down to. For listeners who are kind of newer to the show, I think we've hit on like a lot of stuff that's like very fundamental. And I'm like, I never know when people have started listening to the show, because you can listen to these out of order and you're totally fine for the most part. But the epigenetics is this idea that you have your genetics, which are written down, but like the amount that the genes can be expressed. It's a little bit like a dimmer switch on a light. So like you can turn them on full and it's expressed fully. You can kind of like dim it down. And the thing that is affecting, and so for example, epigenetics has a strong relationship with metabolism and obesity. So people are really interested in it in that respect. So like one of the things that people have looked at is like, how does mom's nutritional status during pregnancy eventually affect the adult health and metabolism of her child? And I think that the idea of doing that for teeth is so cool.
There's a lot of potential. So I have some colleagues who are working on frailty and survivability, and we're talking about trying to integrate the teeth and the fact that dental fluctuating asymmetry. So fluctuating asymmetry is the difference between the two sides of the tooth. You would expect in an individual who's growing without disruptions like malnutrition or disease to have the same size tooth on either side. But if one is a little bit different size, one's bigger, one's smaller, then that indicates that there might be some kind of environmental disruption happening.
When you say different sides, do you mean like your left
Right.
and your right? Okay, but not like inside the same tooth. It's like your left tooth and your right tooth.
Correct. Yes.
Got
Like
it.
the left side and the right side of this. Like the you're looking at first molars, is the first molar on the right the same as the first molar on the left hand side, the size of it? Yeah.
I had a moment of panic where I was like, what if you can make your teeth different sizes? And then I was like, no, you definitely need more caffeine.
There are different sizes in your first, second, and third molars, but that is also, they just progressively usually get
Oh
smaller.
yeah.
It's unique that your third molars are as big, if not bigger, than your first in humans. Some primates, that's a different pattern, but in humans, it goes small or large to small. From your first molar is the largest, then second gets a little smaller, and then your third is the smallest, if it's even there, because some people don't develop their third molars. But so yeah, we're talking about trying to incorporate fluctuating asymmetry, and all the work that I'm doing also suggests that if you have some variation, or you can look at the amount of variation in the cusp spacing, and then that may also indicate some stress or some kind of disruption during development, which then could be one of those indicators that's used in frailty studies and survivability, and in early childhood stress, even in utero childhood stress, because mom's malnourished or is experiencing a disease or something to that effect, then could result in the child having those indicators that maybe influence their adult health and all that.
Yeah. Yeah, totally. Totally. Because I think that especially outside of the scientific community, people somehow think that it's like still a nature versus nurture argument. And I feel like for the most part, biological science has kind of moved away from that and been like, it is nature and nurture. And we just have to kind of figure out like what percentages of each are going into this, because they are intrinsically linked. Like there's no way to pull them apart, really. Well,
Yeah.
that's the science, is trying to figure out like the effects of each one. But like you can't study one without the other.
Yes, absolutely. Absolutely. And that's where, what markers are helpful in figuring out when stress has been experienced and how severe that stress might be. And then to what extent do those changes influence that person's ability to survive to a given age? And that's a huge study in bioarchaeology. It has been for a while, but it's kind of taking on a different life at this point with those indicators and frailty, and looking at different indices of frailty and what that means for the person's adult experience.
Yeah. Sometimes people ask me like, how is this relevant to modern life? And I think that this is like highly relevant to the moment that we're in now, where we're talking a lot about like maternal health in the United States and how our outcomes are extremely poor compared to other countries. Like this has long lasting effects on children, and it can like kind of escalate over generations, so this stuff is relevant to today. I hate when people are like, or I don't hate, but like it always is a good moment for teaching when people are like, this has no relevance. Like, why are you studying dead people? Like, this doesn't matter. It does matter a lot to people who are alive right now.
Yeah. Oh, absolutely. And population history. That's really where, so the study I told you that I'm working on right now, linking LEH2 changes in cusp spacing and then the expression of those accessory cusps. That's the immediate next step that I'm already working on. And then the step after that is to look at other populations and incorporate. So I'm seeking funding to go to Turkey and to Italy and then also Hungary, and looking at those groups and talking about, population history. And that sets up all of this understanding, this longevity of health for 3,000 years in most of these places. That, what are people experiencing today? Is there an explanation for it back a few thousand years ago? Are we seeing the same kinds of trends? What has changed? What hasn't changed? That kind of stuff.
Yeah. Dude, it's so interesting. You can come back and talk to me about this whenever you want.
Perfect.
I do want to be respectful of your time. So we're going to finish up the interview with the same 3 questions that I ask everyone. The first question is, if people would like to know more about the kind of work you do, like what's a good kind of intro to teeth / your work in particular?
Sure. Oh, that's a good question. I mean, of course, the paper that I wrote is a good introduction for what I'm doing, because that's what I'm doing. But, an introduction to how I'm like the foundational texts, they're all amazing, but they're also really technical. What I do is really complicated. But I think a good introduction to what dental anthropologists do and how it's important. So it's this book titled Anthropological Perspectives on Tooth Morphology, Genetics, Evolution, and Variation. It actually has one of the chapters by Shara Bailey and Jean-Jacques Houblin. It's one of the chapters that's called What Does It Mean to Be Dentally Modern? I read that chapter and was like, okay, there's some really interesting ideas going on here. And then there's another paper by Riga and colleagues that looked at a modern, and I don't remember the name off the top of my head, but it's a paper that was published in 2014 that looked at enamel defects and trait expression. And that also was one of the first texts or first articles that I read. And I was like, okay, if I put these ideas together, then where can we go from here? And then that's when I started reading more of Yuka Yernval's work on the patterning cascade model. And then there's more work on cell signaling and why that's so important and how it's all connected. So if someone's interested, I would start with that book and then the paper by Riga. And then if you really want to get more into it, you can read all those experimental biology papers on cell signaling and the physiology, which I just geek out on. So
no, the reason I'm laughing is because like what you just described to me was a patterning cascade system of knowledge about teeth.
That's how my brain is wired now, apparently.
For people who want like a real intro, like pretty close to popular science, but like maybe like a little notch above it, intro text to teeth, I always recommend Tanya Smith's The Tales Teeth Tell. Very alliterative, so I have to say it very carefully. But it has beautiful illustrations and pictures, and it explains like a lot of like the basics really, really well. So I love that book, and Tanya Smith's also really nice. You ever want to talk to her? Yeah, cool. So what are you reading for work right now?
For work right now, I just finished reading a chapter on linear mixed models, actually, because I'm doing some pretty complicated stats for this new paper I'm working on. So I'm reading that, and I'm also more dentally related things. I'm reading papers about frailty because I'm working on a paper with some colleagues on frailty, and I haven't done that as much anymore or as much. And so I'm kind of looking into that foundation piece to figure out how teeth can fit into this idea.
Totally. I think this is the longest I've gone in a year without saying to people, math is really important. And if you want to do anthropology long term, you should study math.
That means most of the people who've been on your show lately are really into the complicated statistics or just,
no, it's just
in general.
I think like listeners actually will write to me and like make fun of me because at least like normally it's like at least every other episode, I'm like, you know what you should do is you should take a statistics class. Statistics is fun. Everyone should do math.
It's true. And understanding, I mean, this is also sort of a geeky moment, but understanding statistics is such an important part in being able to critically read scientific papers. Because there's one of my favorite quotes is there's lies, damn lies in statistics, because you can make stats say anything you want. And so if you don't understand the variables and you don't understand how they're being compared and then what those comparisons mean and how they're being presented, you're not necessarily going to get an accurate view of whether they did it right. And if they didn't do it right, that's changing the narrative.
Yeah, and honestly, it's not just scientific papers. Like people have really pushed in the last, like, I want to say like 20 years when like big data became more widely available. You start seeing numbers in like journalism and public policy and all this stuff. And people frequently misinterpret them.
Yes.
Like a lot.
Yeah. Yep.
Especially when it comes to survey data. It's kind of crazy. So yeah, take statistics.
Agreed.
Third question is, what are you consuming for fun right now?
I am a big Jane Austen fan, and I've read all of her books for a very long time. And so I wanted to try something different. So I found this author, Georgette Hare, who was really active in the 50s and 60s, who has a really similar, she writes about the Regency era, and they're quote romance novels, but it's not like the romance novels today. It's, there's these really strong heroines in these Regency romances that are willing to push the boundaries a little bit. And she's just a really hilarious author. And so it has the feel of Jane Austen, but it's sort of more like, I mean, I guess how I would perceive myself in that, where I wouldn't take any thing from
any
anybody.
guff.
Yes,
yeah.
There you go.
That's an old word.
Yeah, but I feel like it's appropriate for that. So that's what I'm reading right now. I mean, I've read a whole bunch of hers, but I'm rereading the one Arabella right now.
Okay, I'm going to put that on my to read next. Right now I'm reading, have you heard about this book, Careless People?
Mm-mm.
It turns out there's another book also called Careless People, but the one I'm reading right now is by this woman who was the head of global policy at Facebook/Meta back in like 2010. And she's writing about the insanity and corruption and how the corporation got to be so essentially evil.
Yeah.
And every time I sit down to read a chapter, I become incredibly anxious and nauseated, but it's extremely well written.
Yeah.
I'm not that far into the book. I'm at chapter 4, and she's talking about Facebook going into Myanmar. And I don't know if you know a lot about this, but like Facebook dramatically affected what happened in Myanmar and is like potentially, I think, like pretty responsible for some of the things that happened in terms of like the political situation. Anyway, it's
no, I did not know that.
Well, so Facebook tried to get Macmillan to not publish the book.
I'm sure.
So they said it violated her NDA, and Macmillan was like, too bad, so sad. But she hasn't been allowed to promote it as a result. So like, I feel like everyone in the Bay Area is reading it. I'm like, I don't know if anyone else is reading this book, but like this, literally every time I flip the page, I'm like, this is an insane thing that she has just said.
Yeah.
But they couldn't sue. They haven't been able to sue her for defamation because it's all true.
Right. Yeah.
So anyway, sorry. My point, circling back, is what you are reading sounds like the perfect thing to read after I finish this book that is raising my blood pressure.
Yes, and that's why I like to read those books, because I feel like I deal with a lot of heavy stuff at work and in life in general, and so yes, this is a nice escape. So
yeah. Yes. I literally said, what are you consuming for fun? Not like, what are you consuming to really stress you out?
Right. The news would be the answer to that.
Yeah. Damn. I'm so glad we're here in 2025. Well, thank you so much for your time. I really appreciate it. You are welcome back anytime. And I hope you have a wonderful day.
Thank you so much for having me. This has been a lot of fun. And if people have questions, by all means, feel free to reach out to me too. And yeah, I hope to be back on here soon. And if anything else, we'll be in touch later.
Definitely. I'll put a link to your staff page in the show notes.
Sounds good.
All right. Bye, everyone. Thanks for joining me for this episode of the Anthrobiology Podcast. If you have questions or comments, you can reach me at gabby.lapera@anthrobiology.com. That's spelled GABY.LAPERA@anthrobiology.com. This show is produced by me. Music courtesy of Chad Crouch, aka Poddington Bear at Sound of Picture.